Hello, I welcome all of you on your own channel. That's Mudabbir's Biology Club. Dear students, today our topic is the homology in the early development of multicellular animals. Now, in this lecture, we will try to understand that if the animals are totally different, different types of animals are totally different in their organization and other things, why there is a strong homology in their early development. In this case, we will see that all the sexually reproducing animals, they start their life from the male gamete, that's the sperm, and the female gamete. Once this gamete, that's the sperm, fertilizes the ovum, it means the fertilization occurs, the zygote is formed, and the zygote then develops into the morula, and this morula is a solid ball of blastomers. The morula later on develops into the blastula, and the blastula ultimately develops into the gastrula. The gastrula underwent the what we call as morphogenetic movements, and it forms the three germ layers from which all the organs of the body of an animal are derived. So this is the basic pattern of the early development in all multicellular animals which are sexually reproducing. Now, why there is a strong homology in this developmental pattern? It is the point to be understood over here. If the animals, sexually reproducing animals, they are of different organizations, or they are of different what we call as this structural plan, then why there is so much homology in the early embryonic development? It means all the sexually reproducing animals start their life from a zygote, and later on the zygote by the cleavage forms a solid wall of blastomers which we call as morula. For say example, this is the zygote. It is the diploid cell zygote. Then there is this what we call as morula. It is the solid ball of blastomers. You may understand it like this. These are the blastomers, and this is the morula, and the morula then gets changed into the what we call as the blastula. This is the blastula. This blastula has a cavity which we call as blastocule, and then the this blastula ultimately gets changed into the gastrula. This is the gastrula, in which you will see the morphogenetic movements will start, and the morphogenetic movements will establish the three germ layers, that's the ectoderm, endoderm, and mesoderm, from which all the organs of the body are derived. So we conclude over here that this homology in the early embryonic development, in case of these sexually reproducing triploblastic animals, is due to the fact that in the ancient past all these animals used to have a common ancestor. That is, they have evolved from a common ancestor, and due to their common ancestry, there's a strong homology in their early embryonic development. Hope that you have understood well why there is a strong homology in the early development of all sexually reproducing triploblastic animals. This all is due to their common ancestry. For other lectures, do subscribe Mudabbir's Biology Club.